My name's Jacob Prince, and I'm excited to present GLMsingle, a turnkey solution for accurate single-trial fMRI response estimates. In this talk, I'll describe our motivation for developing this tool, the set of optimizations it implements, and then show in 4 different analyses how it's able to substantially improve the quality of GLM estimates in 2 massive fMRI datasets. If you'd like to access code and documentation, feel free to scan the QR at bottom right. Recently, the Natural Scenes Dataset and Bold 5000 have together measured high-resolution brain responses to tens of thousands of naturalistic visual stimuli, offering an unprecedented window into the tuning, topography, and representational architecture of the human visual system. But signal-to-noise ratio is a major challenge in these datasets, given the many possible sources of noise, the existence of so many different conditions to model, and the degree of bold signal overlap between nearby trials. So, to unlock the potential of these datasets, we really need effective methods for modeling brain responses that deal with these different sources of noise flexibly and efficiently, and that's exactly the goal of GLMsingle. The entire pipeline can be run in only a few lines of code, and works on any dataset that has repeated conditions. The function simply requires bold time series data and a design matrix as input, and it seamlessly integrates 3 different optimization procedures to output response estimates in the form of GLM beta weights. First, for each voxel, an optimal hemodynamic response function, or HRF, is identified from a library of 20 candidates by modeling the time series data independently using each one, and then choosing the HRF that has the best fit in terms of variance explained. Then GLM denoise determines a custom set of nuisance regressors to include in the model. This involves running principal components analysis on data from voxels unrelated to the experimental manipulations, and then adding the top principal components one at a time to the GLM until cross-validated variance explained is maximized. And lastly, fractional ridge regression is used to estimate the final GLM betas. This technique uses cross-validation to apply a custom amount of regularization at each voxel, and this is particularly useful for dampening the instabilities in model fitting that can arise from having highly overlapping neural responses to successive trials. By default, these 3 methods run jointly, but users can easily switch any of them on or off, and tune different hyperparameters to suit the needs of their dataset. Now let's see how these techniques affect voxel reliability, patterns of temporal autocorrelation, representational consistency, and single image decodability in the NSD and Bold 5000 datasets. To compute a voxel's reliability, we can simply correlate its responses to odd and even presentations of the same images. For the rest of this talk, I'll be comparing results from 3 different beta versions output by GLMsingle. Version 1 are those from a baseline GLM that uses an assumed canonical HRF. Version 2 have the HRFs optimized at each voxel, and version 3 has HRF fitting, GLM denoise, and fractional ridge regression together. Now looking at visual cortex data from the first NSD subject, we can plot the change in reliability we get from applying GLMsingle compared to the baseline GLM, and we see almost uniform improvement. The scatter plot shows the same data where each point is a voxel, and those above the red line indicate increases in reliability. These trends are broadly consistent looking at the rest of the subjects, with slightly noisier effects in Bold 5000 due to having far fewer repeated stimuli for computing these metrics. And we can summarize these effects by plotting the averages for each beta version, where we see that the different optimizations tend to contribute unique added benefit. And as we progressively look at more and more reliable groups of voxels, the improvements from GLMsingle become more and more pronounced. Another common issue with GLMs is that we see significant correlation between the beta maps from neighboring trials, which presumably reflects poor model fitting. To see whether GLMsingle might help mitigate these correlations in NSD and Bold 5000, we first computed a representational similarity matrix over the sequence of betas from each session, and then took a grand average over sessions and subjects, leaving behind only correlations in time that are unrelated to the content of the randomly ordered stimuli. We can summarize this information with an autocorrelation function, where each point on the x-axis is the average similarity between a given trial and those that are x forward in time from it. So in the baseline GLMs, we see some striking patterns of average autocorrelation that extend several dozen trials forward in time. But GLMsingle does quite a bit to reduce this effect in both datasets, to the point where beta maps from neighboring trials are basically uncorrelated. And this is presumably a good thing for downstream analyses if it means that responses to neighboring trials are being effectively disentangled. Massive datasets like these can be useful for many different representational analyses, whose noise ceilings depend on shared variance across image repetitions, both within and between individuals. So we next asked whether GLMsingle meaningfully affects cross-subject representational similarity. We took responses to the images overlapping among all the subjects and computed RDMs for each beta version, and then we correlated each pair of subject RDMs and observed how these relationships changed from running GLMsingle. And we found clear and consistent improvement in representational similarity, whether we're looking within NSD, within Bold 5000, or between the subjects of the two datasets, whose data were collected on different scanners, at different field strengths, and using different experimental designs. And to go even further, we asked whether GLMsingle helps with the challenging task of decoding individual naturalistic images from neural data in a one versus all context. In both datasets, we found dramatic improvements in linear decoding accuracy, especially in the most reliable voxels of NSD. Altogether, these results highlight how GLMsingle can help improve the quality of existing or future neuroimaging datasets, especially those that sample many experimental conditions. If you'd like to learn more about the methods, they've been written up at length in the NSD data paper. And to access the current version of this tool with documentation and example scripts, you can go here. Feel free to also email me at any time with questions, and thanks for visiting my poster.